Hi everyone, my name is Connor Lane and I'm a student researcher for Dr. Veronica Bruno's Pain and Parkinson's Disease Lab. The present study, which is called Pestering PINK1, was made possible due to her expertise and guidance, as well as the effort and involvement of Parisa Elizadeh, her research coordinator. Parkinson's disease, or PD, is the second most common neurodegenerative disease. PD is a progressive disease that worsens as more brain cells lose their function and die, which traditionally has been studied in relation to the way motor ability and movement worsens. While these motor symptoms are no doubt important to patients, our team is particularly interested in the various non-motor symptoms that occur. Pain is one of the most frequently reported symptoms of PD that can appear even before motor symptoms develop. Anywhere from 68% to 85% of PD patients will experience various types of pain, which deeply impacts their quality of life and can contribute to further difficulty with movement. However, very little is understood about these pain symptoms, as traditional PD medications target the neurotransmitter involved in motor behavior, but are unable to address many non-motor symptoms. Currently, little research addresses pain in PD rodent models, so we wondered if any available rodent model could capture pain symptoms as they do motor symptoms. In our pilot study, we examined the PINK1 genetic rat model, which lacks the PINK1 gene associated with a greater risk for PD in humans. Interestingly, PD patients with a PINK1 mutation report a high prevalence of pain in clinical research, often before motor symptoms develop. We hypothesized that this model would also show early pain without motor dysfunction. In this study, we assessed sensitivity to mechanical stimuli and overall motor behavior in 20 rats, 10 PINK1 model rats and 10 controls. At 3 months old, each rat underwent the von Frey test and the open field test. The von Frey test, applied here for the first time to the PINK1 model, involved poking the hind paw to determine the paw withdrawal threshold, or PWT, which reflects sensitivity to pain. A higher PWT indicates the rat is less sensitive, and understanding sensitivity to mechanical stimulation can help predict pain symptoms. The open field test has many purposes in animal research, but it is often used to assess movement patterns. In our case, there were 3 types of activities being recorded, which includes locomotion, such as walking and running, in-place activities, such as grooming, and no movement with complete stillness. We found 3 important results after testing the PINK1 models and controls. Firstly, the open field test showed that there was no difference for the 3 activities that we used. That means for locomotion, in-place activity, and no movement, the PINK1 model largely behaved the same as the controls. Secondly, the paw withdrawal threshold was significantly lower in the PINK1 model compared to the controls. This means that the PINK1 rats were significantly more sensitive to the mechanical filaments that were used to poke their hind paws. Thirdly, there was no difference in the PWT of the left paw versus the right paw for all of the rats, indicating one side was not more sensitive than the other. There are a few major conclusions that we are able to make from these results. We found that the PINK1 model at 3 months old showed no motor symptoms, making it suitable for early-stage PD research. Importantly, we observed abnormal pain processing in the PINK1 rats at this age, mirroring the early pain symptoms seen in PD patients with dysfunctional PINK1 genes. Both paws were equally affected, indicating the model can capture bilateral disease progression that affects both sides of the brain and body. However, it is important to note that this pilot study is limited because the experimenters knew the identity of each rat. This leaves room for unconscious bias to impact the results, but this is still a meaningful start to the research in this area. Overall, this study suggests the PINK1 model can reflect the clinical observations that occur in the early stages of PD, making it a valuable tool for further research on pain, and ultimately useful for helping to develop treatment to improve the quality of life for PD patients living with pain. I want to extend thank you to the labs of Dr. David Park, Dr. Tuan Tran, and Dr. Patrick Wellen, which includes Cecilia Bradenhorst, for their insight into working with animals and conducting behavioral tests. I'd also like to thank Parisa Elizadeh for her support with animal testing and for her management of the procedural details of this project. Lastly, I'd like to thank Dr. Veronica Bruno for her supervision of this project, which included valuable mentorship and a comprehensive approach to science, as well as for granting me the freedom to select, conduct, analyze, and interpret the experiments presented here. This study was funded by the Calgary Parkinson Research Initiative. Thank you.